Okay. Hey everybody, we're just going to make our video. I don't know what group we are. I think we're group 7, 12, something like that. Less than 12, that's what's important for the homework. So the first one, it is saying, it gives a big old list of a bunch of proteins, sequences, molecules, things that are involved in transcription and or transcription regulation, and it wants you to answer the questions and use that list. The first one says, which term described or which of the following terms describes or an example of regulatory proteins? And those are the activator, the repressor, and the CAP. The next one is, which of the terms describes or is an example of a small effector molecule? So something that affects it. That's an inhibitor, allolactose, inducer, and CAMP, cyclic AMP. C, which of the following describes an example of or is an example, typo, of the sequence found within the DNAs. So which one's a sequence, a DNA sequence? And that's +1, -10 operator, LAC-A and LAC-Z. And then it says, which of the following above is not involved in the LAC operon, and the inhibitor is not, and which of the following is an example of a structural gene, and that's LAC-A and LAC-Z.
Okay, so number 2 is the fill in the blanks. A asks, the LAC operon is regulated by 2 inducers, and those 2 inducers are allolactase and the CAMP. For B, it says the binding site for sigma factor and RNA polymerase is called the LAC, and that's P, while the CAP site is the binding site for the activator that regulates the LAC operon. C, it asks for the enzyme that converts ATP to cyclic AMP, and that's adenyl cyclase. D, it asks the -35, -10, and +1 sequences are found within the LAC-P portion of the LAC operon. E asks the blank gene encodes the LAC repressor while the blank gene encodes lactose permease. And the LAC-L is what encodes, and the LAC-Y is what encodes the lactose, the gene. For F, it asks, blank can regulate gene expression in bacteria by causing transcription to terminate prematurely, and that's the attenuation. G, it says an E. coli strain that contains 2 copies of the LAC operon is called the merozygote strain.
All right, for number 3, we were figuring out what will turn blue and what will not turn blue. Basically, it says, supposing you're examining the LAC operon in E. coli, in your experiments, the activity of the enzyme B-galactosidase is measured by excluding X-gal and IPTG in the growth media. ITPG can mimic allolactose. IPTG can also bind to the LAC repressor and release the repressor from the operator. X-gal is a molecule that is a substrate for B-galactosidase, and it turns E. coli cells blue when metabolized by beta-galactase. And it says, answer the questions part below, determining the culture conditions in A through E result in blue E. coli cells. So it says, for this problem, assume that E. coli strain is wild type. Okay. And ATP is abundant. So it says, ITPG is absent, glucose is present, and L-gal is present. So for this situation, it will not turn the cells blue, and that's because if ITG is present, and it will bind to the lacto. If glucose is absent, the cyclic AMP levels will increase and be really high, and cyclic AMP will bind to the CAP, and the CAP bind site. For B, it says IPTG is present, glucose is absent, and X-gal is present. This will result in blue cells. So kind of the needed thing is IPTG and X-gal are the 2 things that are needed to result in blue. Yeah. So since knowing that, for the next one, if X-gal is absent but ITG is present and glucose is absent, it's also not going to result in blue cells. B, IPTG is present, glucose is present, and X-gal is present. You would think, okay, if those 2 are present, that's going to cause it to be blue, but that's not true because when glucose is present, the cyclic AMP levels will be really low, and they won't be able to bind to the CAP, which will inactivate and cause it not to be blue. I think there's only, oh, then E. E is the last one. So it's APTG. IPT, it's too many letters, is present, glucose is not, and NACTH is absent. Kind of the same thing. The cyclic AMP levels will be low because of the glucose, so it won't bind. And the lac operon won't transcribe, so it won't be blue.
Okay, number 4, it says, below are the lac operon genotypes of several E. coli strains. Please note the following. So it just explains the whole minus and the L and the plus and whatnot. And so it says, do these E. coli strains display constitutive, inducible, or non-inducible expression of the LAC-Z gene? So the first one, it's I plus, P plus, O plus, Z plus. And this one is going to be inducible because in the absence of lactose, the LAC repressor is going to be bound to the LAC-O and the lac operon transcription, which means that the transcription will not occur. So that's going to be inducible. For B, we have I subscript S, P plus, O plus, Z plus. And this E. coli strand is non-inducible because the LAC-IS allele produces a repressor that is going to remain bound to LAC-O, either in the presence or not in the presence of lactose. So that's going to mean that that's non-inducible. For C, it's I positive, P minus, O, C, Z plus chromosome over a I plus, P plus, O, C, Z plus, F plasmid. And this is going to be constitutive because the copy of the lac operon in the chromosome contains a defective promoter sequence. And so that's not going to be recognized by the RNA polymerase, the plus sigma factor. For D, we have the I plus, P plus, O plus, Z plus chromosome over the IS, P plus, O plus, Z plus, F plasmid. And this is going to be non-inducible, kind of like the one before. The LAC-IS allele on the plasmid produces diffusible LAC repressor protein, and that's going to bind to the LOC. And the LAC-IS is going to remain bound to both copies of the LAC-O. And E, last one, is the IS, P plus, O plus, Z plus chromosome over the I plus, P plus, OC, Z plus, F plasmid. And this is going to be constitutive as well because the IS allele on the chromosome produces a lac repressor that remains bound to the chromosome LAC-O allele. In the presence or not in the presence of lactose.
All right, sweet. For number 5. Number 5's got a big old thing. I'm going to try to screen share and see what happens. I don't know how to do this really. Let's figure this out. Entire screen. Okay, share. I'm going to share this. And hopefully it's still working. So we're going to jump down here so you can see our chart. So yeah, let's just read through this. It says the question concerns the Jacob, this experiment. I can't read lately. Jacob, Monod, and Pardee experiment that utilizes the mutant and merozygote strands of E. coli to examine the lac operon. Suppose Jacob, Monod, and Pardee uses strands listed in A through E. Below the experiments recall, blah, blah, blah. The 2 tubes, 1 tube does not contain lactose, while lactose is added to the other tubes. Your job is in the problem to figure out how much yellow product will be in the presence for each tube. So use the following scale to fill up the table. 0 indicates no yellow color. 1 indicates the amount of yellow color when 1 copy lac operon is transcribed. 2 indicates the amount if 2 were transcribed. So the little dashes, the dash in the table refers to the defective gene, for example, refers to defective lac. So dash is no, activated, plus means wild type. C, lac repressor cannot bind to O. OC. IS produces the lac repressor that can bind to the repressor but can't bind to the allolactose for this problem. So for A, B, C, D, and E, you're just going to read through each one of the genotypes, kind of like the last problem. So I'm not going to read through them. As you can see in my gene, this is the 1. So if we have that, we're going to get 0 color in the plus lactose, the 1 file that has lactose, and 0 that doesn't have lactose. And that's because it's negative. The I negative right here is going to interfere. The next one, we're going to get a 1 strand of yellow and 1 strand of negative. For the third, with the plasmid, trying to remember what's different. Oh yeah, the O's are the different in the plasmid. So when it's O, it refers to a defective operator sequence. And but since that's just in the plasmid, we're going to get 2 strands in the positive and then 1 strand in here because of that. For D, it is got a negative right here. So that would mean without the lactose, it's not going to work. But with the lactose, we're going to get 1 because of the plasmid. And then right here, this one's got the negative. So we're going to get none again without. And then since it's got the IS, produces the rack repressor that cannot bind to the allolactose. Since this one can't bind as well, we're also going to get 0. Let's figure out how to get back to where we're stopping. Oh, I think we're back. Okay, so that was kind of it. I hope you guys understood. This one was kind of confusing, but there you go.